All right. Hello, everyone, and welcome to our third annual webinar on European Foulbrood.
So yeah, we'll just start with introduction. So my name is Peter Fowler. I like to remind my beekeeping audiences that I am not a beekeeper. I am a veterinarian. Uh, I'm currently a postdoctoral research associate in the Milbrath Lab at Michigan State University, Department of Entomology, and my focus is European Foulbrood, which is what I spent my PhD studying. Um, I originally did my undergrad at University of Washington in Seattle. I moved out to Michigan for vet school. During vet school, focused on public health, One Health, disease ecology. Graduated from vet school in 2020, and then continued on. The vet school has this program called Comparative Medicine and Integrated Biology that's meant to foster veterinarians into kind of the world of academia. And so I joined this PhD program right out of vet school. Originally worked with ticks for a little while with Dr. Jean Sao, which was awesome. But then I moved on and did my dissertation on honeybee diseases, specifically European foul brood, which I'll be talking at length about today. So,
And I'm Meghan Milbrath. I'm currently an assistant professor in the Department of Entomology and in the Department of Large Animal Clinical Sciences at Michigan State University. And um, I did my undergraduate in biology at St. Olaf College in Minnesota and a master's of public health at Tulane University and my PhD at the University of Michigan. And all of that was in public health focus and disease transmission, risk models, and epidemiology. So both Peter and I are coming to the honeybee world with kind of a disease background, really. And so during that, I worked on complex systems and the interdisciplinary program on infectious diseases. And I started to do bees, I almost said competitively, for research for my postdocs. I worked with Zachary Huang looking at Nosema co-infection. So that was at Michigan State. So that's how I moved to MSU and started doing research on bees. I also spent a year recently at Swedish Agricultural University working on honeybee diseases there. And I am a beekeeper. I started keeping bees as a child in the 1990s in northern Wisconsin, just as a hobby with my dad. And then I've been more serious about it since about 2010, and I run a beekeeping business. And I'm a sideliner beekeeper with usually between 50 and 100-ish hives. Um, in South Central Michigan. So, thank you for joining us tonight. We really, really appreciate it that people take the time to learn about honeybees and their diseases. We're going to start with Peter's going to introduce our latest research on what we know about EFB and really highlighting some things that we've learned the last year. We're going to then go into talking about practices for managing European fall brood. And the whole time, we will be taking questions. I'll be watching them and answering them as they see fit. So don't hesitate to ask them. If you do want to ask a question, this is in the webinar format, so you'll want to go to your toolbar in Zoom and go to the little Q&A button, and then I'll see the whole list of them and then we'll kind of answer them as we get to them. So hopefully everyone can find that and can ask questions throughout, and then we'll also have lots of time at the end. If you have questions later after this webinar, we have a system set up on our website through Ask Extension. So if you go to our main website, which is pollinators.msu.edu, and we'll put some of these links in the chat, there is a form that you can use where you can upload a question. And it doesn't even have to be about EFB or bees or anything. You can put that in there. The reason that we really like it is that it gives us reminders that we have to answer them. We can assign them to different people if there's someone else who can better answer them. And you can also upload photos. So it's really nice if you do suspect you have European foulbrood, you can put some photos there and ask through the Ask Extension system. And then if you do get bored and you want to leave and watch us on double time later, Or if you do just want to revisit some things, we will post this video after we do the accessibility things on YouTube to our Michigan State University beekeeping channel. We also do have the last 2 years videos on this. So like I said, this is our third annual EFB webinar. And so you can go back and see the other ones. I mean, obviously there'll be a lot of overlap. I wish we had an hour worth of content every year and we were making such great progress, but there will be some overlap, but we do try to mix it up and have new information each year. One of the things that is new this year is we do have a new online tool called Diagnostic Tools for Honeybee Diseases. So I'll have a QR code up for it later, but you can also access it from our pollinators.msu.edu site. It has visual signs of disease, so you can see the listing of all the diseases there. It gives you step by step through the field tests, including like the lateral flow devices, alcohol washes, microscopy. And then in each of those step by steps, it will tell you when you should do the test, what types of samples you should take, common mistakes that people do, which is my favorite part because I feel like that isn't listed a lot, and then how you would interpret the results. So that was funded under some grants from the USDA Veterinary Services Training Grant and through the SARE Research and Extension Program. And so you can also access it through beesneedvets.com. And I'll put these in the chat when I get done talking. And there's the QR code. And it should work on your phone as well. And here's just an example. There's games on there too, which I think are particularly fun. But you can see what's happening in this image, and then you click that it is American Foulbrood, and then it will kind of walk you through. So you can actually test yourself on what types of samples to take and how you can identify diseases visually. And here's an example from the blacklight test. So there's diagrams and hopefully this works, and videos. Peter made this lovely video of how a blacklight works to show AFB scale. So, all right. And so now we're going to get into the meat and I'm going to hand it off to Peter.
All right. So, in previous webinars, I've gone through the history, and folks should look into the history, like reference those webinars if they're curious. Today, I'm just going to like give a broad overview of like the current state of knowledge of European Foulbrood, and then I'm going to spend most of today talking about like the research we've done in the last couple of years at Michigan State University, with kind of a focus on the day-to-day and what the actual research looks like behind the scenes. So hopefully you find that interesting. So I'm going to start off with what do we know about European Foulbrood. Now, I like to do this in the context of what we know about American Foulbrood, because I think this is the disease that everyone is more familiar with. So there's two bacterial brood diseases that affect honeybees. American Foulbrood is the one that's more well known, and there's a couple of reasons I think that is. We know a lot about the bacteria that causes American Foulbrood. So it's one of over 200 different Pannibacillus species. It's called Pannibacillus larvae. So we know a lot about it, where it came from. It's a rod-shaped bacteria that forms these hardy spores that can last for decades in the environment. We know it persists on equipment for a very long time. One of the reasons this disease has been so well researched is because once you see signs for American Foulbrood, it's almost always fatal. So for that reason, beekeepers tend to take it pretty seriously. They burn their equipment, and there's 3 different antibiotics that are approved for this disease as well. And I think because there's 3 treatment options and because people take this disease seriously, it's largely come under control in recent years. And we'll look at some of that data in a bit. So in contrast, European Foulbrood is caused by this really, really unique bacteria. So like I said, Pannibacillus larvae is one of over 200 different Pannibacillus species. This bacteria that causes European Foulbrood is known as a single genus, single species bacteria. So It's the only Plutonius is the species. It's the only species within the genus Melissococcus. So it's very, very unique, and we aren't sure how it evolved to fill the niche that it fills. Um, it's very difficult to work with in the lab, which I'll talk about a little later. But one of the reasons this disease in general is so poorly studied, beyond this bacteria being difficult to grow in the lab, is that the hives tend to get better later in the season. And we're not entirely sure why that is. Uh, but the picture also seems to be changing a bit, and the disease seems to be lingering longer. Uh, so because the hives survive, people have generally ignored this disease. There's still only one treatment that's approved to treat this disease, oxytetracycline, and no one has tested to see if strains in the United States have become resistant or not, which is a surprise to me when I started studying this as a veterinarian. So a brief description of the disease. So this is the bacteria Melissococcus plutonius under a microscope. The disease starts, so this is what you expect to see with a healthy honeybee larva. It's nice and pearly white. When the larvae get fed feed that's contaminated with this bacteria, they start to discolor and become kind of clear. And you can see uh, this is almost transparent. You can see these white lines here. This is the tracheal system of the larva. So this becomes apparent in early signs of the infection. At this stage, when it's showing these symptoms, if you did a cross section, and this is basically a cross section of the larva that was taken as part of a study done by the Woods Lab up in Canada, and all this purple stuff in here. So this is the midgut compartment, this big cavity here, and all this purple stuff in here is the bacterium Melissococcus plutonius. So it's pretty much filled up the entire midgut at this point. The larva starts to twist within its cell pretty often. It'll form this kind of corkscrew with its head pointing out. And then after the larva actually dies, Melissococcus seems to kind of sink into the background, and other bacteria tend to take over the dead larva's decaying mass, and they exhibit all these different weird symptoms. So some of them will look like pinkish, some of them will be kind of watery yellow, some of them will look like kind of crusty. And we're still completely unclear why all these different symptoms occur. Maybe it has something to do with the secondary bacteria that's present, but it hasn't really been studied extensively. We do know Melissococcus plutonius persists in these samples, even though a lot of other bacteria tends to be present. And eventually, if this material is not removed by the adult nurse bees, the mass will kind of dry down into this scale that's usually kind of rubbery and loosely adhered to the back of the cell. And Melissococcus plutonius is also found to persist inside these scales. So we'll talk about persistence a little later. We also know, based on research that's recently been done in Japan, that there's two distinct lineages of this bacterium, Melissococcus plutonius. So one's commonly referred to in the literature as typical, and the other one is referred to as atypical. And they vary quite a bit in their characteristics, both in the laboratory and what they do to larvae. These typical ones, sometimes they cause really, really bad disease. Sometimes you can give this bacteria to larvae and it does nothing at all. The atypical ones, on the other hand, almost always cause disease in the larva. And how they study that, this is from this same group in Japan, is through the use of this in vitro larval assay, they call it. So basically, they graft developing larvae into these little wells, and then they grow them in an incubator in the lab on an artificial diet. And that allows them to feed these larvae different strains of the bacteria that they've purified and see what the differences are. So in this test here, you see this top row has not received any bacteria. So this was their control group. The middle one here received one of these typical strains, and you can see it looks pretty much the same as the one that didn't receive any bacteria, not really any difference. The one on the bottom, all these ones received this atypical strain, and you can see pretty much all of them died. And so that produces what they call a survival curve, which is this graph here. And it's kind of the percentage of larvae that are surviving at each day. And you can see control and one of the typical strains are on top, and this atypical that they tested it against killed all the larvae by the fifth day. So it's pretty virulent in the lab. So that same group also did these cross sections of the larva during different stages of infection, which is really interesting because we're still unclear how it actually kills the larva, but I think this gives us some clues what they've done here. So in early infection, and they've done this for quite a while, the bacteria seems to grow in close association with this thick layer that protects the larva's midgut. So this is another cross section. This is the inside of the larva's gut, and this is the kind of lining. And this thick layer between the purple and the red here is called the peritrophic matrix. And this is a normal structure in a larva's midgut, and it's kind of a thick chitinous layer that's important both for digestion and to protect the larva's midgut from pathogens. So you can see this, it starts growing in association with this peritrophic matrix. It continues, and then in the next stage of infection, it starts to break down the peritrophic matrix. So you can see Melissococcus platonis has all the red stuff here. It's pretty much taken up the entire midgut like we saw earlier. But the peritrophic matrix is gone in these later stages, and this may underlie some of the symptoms that we're seeing. But we're still doing more research on what the actual mechanism, how it's actually degrading that matrix is unclear. Okay, so that's what we know about pathogenesis. What do we know about prevalence of this disease? So what we're looking at here is data that has been collected by the USDA lab in Beltsville. So they've been collecting samples from beekeepers for more than 100 years. Beekeepers can send samples that they think might be diseased, and USDA lab will diagnose the sample. They'll look at it under the microscope, see what bacteria is present, and they'll determine if it's either American foul brood or European foul brood. And if it's American foul brood, they'll test for resistance, which I'll show you a little bit later. But here we have a year by year breakdown dating back to 1985 of all their data. And basically, this is percent of samples they received each year that's positive for each of these diseases. So the gray line is American foul brood, and the red line's European foul brood. You can see American foul brood was super common. Over half of the colonies for a very long time had this really, really bad, severe disease. And there was a lot of control efforts in the early 2000s to get this under control. And this is also around when they approved two more antibiotics, lincomycin and tylosin, to treat this disease. So I think a combination of education, eradication efforts, and treatment strategies have really brought this disease under control in recent years. But we see kind of an opposite picture with European foul brood. This red line you can see started to kind of creep up starting in 2005. And in the last year, it's been, a record high, over 30% of the colonies in 2022. So we're not sure if these numbers reflect reality. So this is what we would call passive surveillance. Because people suspect there's a disease in their hive already and they're sending something in, we would expect this to overestimate the numbers. But based on our research in Michigan, we think this might be pretty accurate. And I'll go through one of the studies that kind of cued us into this now. So there's long been this story about European foul brood that has something to do with hives that are in blueberry pollination. And we hear this over and over and over again from commercial beekeepers that, this in different regions of the United States, even that when hives go out to blueberries, when they come out of blueberry pollination, they seem to get really, really bad EFB. And so it's been suspected that something in blueberry pollination might be triggering this disease to manifest. And so that's what caused us to undertake this experiment. Basically, we took 60 colonies from one of the commercial beekeepers and we split them into two groups. 30 of them went to pollinate blueberries, 30 of them went off to a distant holding yard. And then we monitored them over the course of the season. So we had three different time points when we kind of followed up and checked for prevalence of this disease. And in this top group, the one in blueberry pollination, we found 62% of these hives got EFB. And on the bottom, the one that was in the holding yard, we found also a pretty much identical percentage of hives got EFB. And the main takeaway was that this is a huge percentage. Like 62% of the hives getting this disease is really, really bad, right? And what compounds that is that we found that the hives that got severe EFB, so each of these dots is from this study, one of the colonies that was severely diseased with this EFB. And on the y-axis, you have the percentage of growth over the course of the season. And so we got diseased hives on the left and healthy on the right. And we found that the ones that had disease, even though they could struggle through the season, they had a 54% reduction in their growth. So with the prevalence, this might be having a pretty big untold impact on our commercial honeybee colonies and maybe even our backyard ones, right? So when I started my PhD, we kind of knew that this was a problem in Michigan, but we didn't know how widespread it was. So what I wanted to do is determine, is this problem extending into sideliners? Is it also a problem for hobby beekeepers? Is it a problem in all the commercial operations? We didn't know how broad the problem was. So we undertook active surveillance. So this was a 2-year cross-sectional surveillance project. So what that means is we were actively looking for the disease in the landscape. And in this project, we included all the areas you see here in gray are represented. 6 commercial beekeepers. We had 7 sideliners. So sideliners are smaller beekeepers. I think we defined it as between 100 and 500 hives or so. And then hobby beekeepers, beekeepers that keep bees in their backyard, maybe 50 hives or less or so. Um, so as part of the surveillance, we would go out to their apiaries. We would select hives at random within the apiary. And then we would go down to the brood nest and look for clinical signs of EFB. So this is kind of some of the signs, and we'll go over some of these a little later in the talk also. So that would give us the prevalence within those operations. And then we would take larval samples, whether the hive was sick or not, and we would collect them into this little bag called a whirl pack, take them back to the lab. And then we made these little kits that we sent off to hobby beekeepers so they could do the same, send us larval samples, and we could see if we could find the bacteria in their hives. So back at the lab, we would have these little whirl packs. So this is 10 larvae and 10 mils of phosphate buffered saline. And the first step we do when we get them back to the lab is we smash them up and make kind of a larval soup. And the point of this is to basically break open the larva's midguts. So all that bacteria, if it's in there, will be now out floating in this buffer. And then we take a tiniest drop of that and we put it in a small tube called a microcentrifuge tube. And then we put it in this block and heat it up. And basically at this point, we're boiling the water in order to break open those bacterial cells. So if there's the Melissococcus plutonius is in these samples, we're hoping it busts open and then the DNA is released into the liquid. And the DNA is really the target of our screening and what we're looking for in the next step, which is PCR. So we're all familiar with PCR because COVID testing, it's the super sensitive test that can detect COVID virus at very, very low levels. But I wanted to go through what it actually looks like for us to run this in the lab, because even before I started doing this, it was kind of abstract to me. So this is how we run PCR on our larval samples. We make a concoction of a few different ingredients. So the first thing is this commercially available thing called a master mix. That's this green stuff here. And this has all the components you need to replicate DNA, basically. And then the other 2 components are called primers. And these are little chunks of DNA that are specific to a gene that's only present in our bacteria, but isn't present in any other bacteria. So we choose chunks of the gene that are different sizes, and that'll come into play a little later. So there's a company you can order these little segments from. We add these also into this master mix, and then we aliquot this into all these little tiny vials. So each of these vials at this point is labeled for a different hive, but it all has the same stuff in it. Master mix and our primers for these 2 chunks of DNA, one specific to typical, one specific to atypical. So we then will add the tiniest drop from our boiled larval soup. 2 microliters we put in here. So we put it in the corresponding tube, and then we put all these tubes in this thing called a thermocycler. And what this thing does is basically heats up and cools down the mixture. And each time it does that, it melts the DNA. So the DNA, 2 DNA parts come apart, and then these probes can kind of stick on there. And then as it comes back together, it'll replicate. So as it cools, it'll melt the DNA and replicate it with each cycle. So where 1 copy was in your original sample, now you have 2 copies. And if it does that enough times, it turns 1 copy into billions and billions of copies. So how is this useful? Now we have billions of copies of a little chunk of DNA, right? Next thing we do is called gel electrophoresis. So this lets us separate these chunks of DNA based on their size. So we carefully load. This is a very tedious process, and I want to express the extent to which our myself and our lab tech have struggled in the lab doing these day in and day out with hundreds and hundreds of samples. But basically, you load the sample that comes off of that into this. This is kind of a gelatin that you cast, and it has these holes in it. And you load your sample into there. And sometimes it doesn't load. Sometimes you tear the gel and you have to redo it. A lot can go wrong at this stage. But the idea is you run an electric current through it. And the DNA, because it's got a negative charge, will travel through this gel. And based on the size of the chunks of the DNA, it'll travel at different speeds. So the smaller ones will move really fast, the slower ones will move really slow. And then we can put it under UV light and look at this for presence of the DNA. So each of these bands is a chunk of DNA. So to give you an example of what we're looking at here, this is one of the wells where we loaded one of these samples from a hive. We ran the electrophoresis, and then this band is higher up, it went slower. This band is lower, so it went faster, smaller, so it went faster. So within this hive, within this little band here is billions of copies of that chunk that was copied from our atypical DNA. So this would be a positive for atypical and typical, both within the same hive. You can see these ones were negative. There's no bands here. You can see how many are positive already. And you can also see that a lot of these hives have both atypical and typical within the same hive, pretty much across the board. And we'll look at this data in a second. So now we've gone out into the field and we've established prevalence of disease, and then we've established prevalence of infection, which is this bacteria was present in our samples, right? Next, we want to ask more questions of this bacteria, though, right? So we need to isolate the bacteria from these samples. So for each of these samples where we had a screening that was positive, we would culture and purify Mosacoccus plutonius. And this is what that looks like. So Mosacoccus plutonius is super finicky to grow in the lab. This is me working in an anaerobic chamber. It doesn't like growing in the presence of oxygen. So within this anaerobic chamber where we've vacuumed out all the oxygen, we'll take a little bit of that larval soup and we streak it on this agar plate, and we dilute it by streaking it more and more in different what we call quadrants. And then we put it in an incubator and incubate it. And after it gets out of the incubator, if there's bacteria present, any bacteria at all really, that can grow anaerobically, we'll see something like this. And so this top part of the plate is where I started to streak. You can see it's really dense. A lot of stuff grew. It gets more dilute as I go across the plate. And then eventually we end up at a spot like this where you see these little tiny dots. And the idea behind this is you dilute it so much that eventually just a single bacterial cell will be present. And eventually when it grows, it clones itself and it clones itself again until there's millions of copies in this little single dot of just what started as one bacteria. And that allows us to isolate the bacteria and purify it. So we know we just have one bacteria we're working with. So I'll take a sterile toothpick, I'll touch this colony here, transfer it into a little bit of broth, and then we can grow it up and then freeze it and do a bunch of experiments with it later. So today, we've done this with hundreds and hundreds of different Mosacoccus plutonius isolates that we have that we've been studying. So, all right. So that's the process of what we go through in the lab, what I've been doing for the last few years. What did I find? How widespread is EFB? And again, this is kind of the area we were covering. We were looking for signs, infection, and then we were cultivating the bacteria. And I'll go through what we did with that cultivated bacteria in a second here. So 855 colonies were included in this cross-sectional portion of this analysis. And we found about the same thing that the USDA found, that 1 in 3 colonies, 33%, were infected. So the bacteria was found in those colonies. And this was present across all sorts of beekeeping operations. There wasn't any beekeeper where we sampled more than 2 hives where they didn't have at least some bacteria, Melissococcus plutonius, present in one of their hives. And what's more, half the ones that we found infected with this bacteria had both typical and atypical present in the same hive. So these co-infections, we're not really sure what they mean, if they're important. It's kind of the next thing I'm kind of interested in. And we didn't really see any geographic spread to it either. Like they seem to be prevalent across the entire range of our surveillance. So all the blue areas on this map here are areas where we found typical and atypical. And there were the 2 gray counties here. These are just the counties that we had like 1 hive from a hobby beekeeper in each of these counties. So like it's not really representative. But everywhere we sampled sufficiently, we found both these strains were present. So present pretty much across the entire range of our sampling area. We also followed commercial honeybee colonies out to almond pollination. So this is something pretty much all commercial honeybee colonies participate in every year, is going out to California to help pollinate almonds, which need honeybees to pollinate. So we went out to California to see if this disease and infection was present out in California. And then we followed a bunch of colonies also to Georgia in April. And then the rest of this data is just a breakdown of how much both the light blue is just infection, and then the purple is the percentage of colonies that we sampled that month that had the clinical disease. So you have kind of a demonstration of the seasonality of this disease, that it gets worse and worse in the spring. It peaks in June, and then it kind of tapers off, like people have mentioned. It seems to resolve. So there's a couple things to point out here. First off is that June is awful. Like 1 in 5 hives were sick with this disease, and over 50% were infected. This is like a huge rate of disease. So very bad news, right? Additionally, there's a lot of infected hives that are not clinical. So this makes this very tough to manage a disease like this, where you have a lot of asymptomatic carriers of the disease. And once the disease starts resolving, even though the clinical manifestation of the disease is gone later in the season, we still have really high rates of infection. So yeah, this is not good. So the next question was, what do we do about it? Like, this is a severe problem. It's across all sorts of operations, commercial, sideline, or hobby beekeepers. One of the things we tested pretty early on was to see if this behavior called hygienic behavior is effective. I'll say right off, it isn't. But what this is a behavior by which honeybees will remove dead brood from the hive. So it's kind of a test to see how fast they can do this. So you kill a chunk of brood. That's what we're doing with these little tubes here. So we put one of these PVC pipe tubes over a chunk of brood comb, and then you pour liquid nitrogen in it. It kills all the capped brood underneath there, and then you return it to the hive, and then you check 24 hours later, and you can assign a hygienic score to that hive, like either it removed, 80% or 90%, or so that gives us the method of kind of quantifying the hygienic behavior of a hive. And we found that there was no real benefit to hygienic hives. They both got EFB at the same rate and seemed to do just as poorly. So that was kind of disappointing. The next question for my thesis, though, was about oxytetracycline. Like I mentioned, we've been using this antibiotic for 70 years, and nobody has tested in the United States, at least, to see if these strains of Melissococcus are resistant to it yet. So I want to start off real quick by describing what this antibiotic is doing because it's important for our story a little bit later. So this is a bacterial cell, and within bacterial cells, you have these things called ribosomes, and these are important for turning RNA into protein. So this is a critical function when the bacteria is growing. When you give oxytetracycline, it makes its way into the bacterial cell, and then it binds to that ribosome and keeps it from replicating the RNA. So the RNA is just kind of stuck there. So it doesn't kill the bacteria, but it does keep it from growing. And we call these antibiotics bacteriostatic. So the cell can't divide, but it's not necessarily dead. A couple of ways that are common for bacteria to become resistant to these antibiotics. There's many, many different ways, but these are two of the common ones. It can acquire a mutation in the ribosome, so the ribosome itself can change shape, and then oxytetracycline can no longer bind there, and then the bacteria can continue to make protein as if the antibiotic's not even there. Another common way is called efflux, so it can acquire genes that can allow it to put these pores or these pumps within its cell membrane, and then when the antibiotic comes in, it can shoot the antibiotic back outside the cell. So this would be called efflux. So usually you can find traits for these in the bacterial genome, and we have not found traits for this in the Molluscoccus genome yet. So we suspect it may be resistant by mechanisms that aren't included in kind of the common lexicon for resistance. But we'll go through how we test for this in a second. So we've been using this for 70 years. We've known for a long time that American Foulbrood Pannibacillus larva has been resistant to oxytetracycline. For EFB, we have not tested yet. So this is how we actually test to see if a strain of this bacteria is still susceptible to oxytetracycline. So what we do is basically we make a bunch of different media plates with differing concentrations of oxytetracycline. So each of these is an identical plate with increasing concentration of this antibiotic, right? And then we spot the bacteria, so a purified strain of our bacteria of interest, onto each of these plates, and then we incubate them, and we see at what concentration the bacteria is inhibited, right? So the threshold that has been established is if the bacteria grows on all these plates all the way up to 8, we consider this bacteria to be resistant to oxytetracycline. So to put these numbers in context, I know they're kind of arbitrary right now. If you feed a hive about 1.5 times the normal dose, so 300 milligrams of oxytetracycline, and you check the larva 24 hours later, you'll find about 12.6 micrograms per mil of oxytetracycline. So if you fed it a standard dose, which would be a little bit lower, right? 200 is the normal dose. We would expect bacteria that survives here to also survive here. So that's kind of where those numbers come from. So we decided to do this with, I mentioned we have collected over 300 strains of this bacteria now, and we decided to do this on a subset of them. And this is what one of these assays looks like. So we have increasing antibiotic going from left to right, and then down here, 16, 32, 64. And you can see this is after a couple days in the incubator. We have growth on these plates that don't have any antibiotics, but even at a very low concentration, we have this bacteria is inhibited. If we wait a few more days, one thing I didn't mention, oxytetracycline is it's not very stable in the environment. It breaks down very quickly. So as we keep these in the incubator, the antibiotics breaking down over time. And we can see the bacteria starting to grow at these higher and higher rates, or these higher and higher concentrations, but we still don't see any growth on 8. So we don't see any evidence of what we would call resistant strains of Molluscoccus plutonius. But like I said, this antibiotic's not killing the bacteria, right? So maybe the bacteria is surviving. What happens if we leave these plates in the incubator until all the antibiotics worn off? So I check these 3 weeks later, and this is what we come back to. So the bacteria is alive, and after the antibiotic wears off, it just starts growing again. So it's persisting beyond the efficacy of the antibiotics. Even at these very high concentrations, a lot of these strains are still persisting. So what does this mean? Is it also persisting on our frames, on our comb? Is it on our suits? Do we have to worry about it like we do American Foulbrood? Is it also on our hive tools? Is it hanging out in the environment for a long time? So this was a question that was asked by this person here, Robin Howley. So she was a veterinary research student with us a couple of years ago, and her project involved inoculating different surfaces. So these little bags here were all surfaces. So she inoculated honey, wax foundation, wood, steel washers, cloth, um, materials that you would find around an apiary, inoculated them with the bacteria, and then we would culture them at regular intervals to see if the bacteria was still alive. And she found 2 weeks later that it was surviving on all these surfaces. So this is some of the bacteria after 1 week, after 48 hours. Atypical strains survived 2 weeks, and she left us a longer term sample after she left, and we regrew this from these surfaces 6 months later as well. We don't have any longer data than that, but we do have, so all the atypical strains remain viable on surfaces. What does it say? Hold on, it says a year after. Um, okay, so all these atypical strains remained viable on all surfaces a year after at refrigeration temperatures. So she did leave us a longer term one after 6 months in the refrigerator, and we recultured that a year and a half later. So that hangs out pretty long time at refrigeration temperatures. And this is a video of the bacteria. Let me see if I can get this to play again. So it forms these aggregates that we saw under the microscope, and the bacteria is kind of sitting within these kind of weird structures, and we were really sure what to make of this. What's more is when I first started sampling in 2021, I would make these larval smears, and this is basically you take a sick larva and you smear it on a glass slide, and then we put these in these slide boxes, and we just left them in a drawer in the lab. And I was able to reculture from these larvae that were just hanging out in a drawer in the lab after 3.5 years. So this bacteria probably lasts a lot longer than we realize. And the next thing we wanted to do was look at how it's persisting. So 3.5 years is a very long time for a bacteria that doesn't form a spore to persist in the environment. And we suspected that it was probably making biofilms. So this is electron scanning microscopy of Melissococcus plutonius after it's been inoculated onto a wood surface. So these are the bacterial cells from the atypical strain. And then these two panels here are from the typical strain. You can see already they physically look very different, these bacteria, between typical and atypical. And they seem to be doing different things to persist also. The atypical seems to be shooting out these strings that are attaching to the surface. And there doesn't seem to be that same behavior going on with the typicals at this point. So we did this again after we let the bacteria dry down onto the surface for 24 hours. And we found quite a bit of this extracellular matrix, which we think is probably biofilm. So this would be something that the bacteria exudes out of the surface of its cell and then uses that as a protective layer to protect it from environmental stresses, disinfectants, antibiotics, exposure. So you can see all this stringy stuff here is covering the bacteria cells are underneath here. So this is one of the bacterial cells here. This is kind of the typical one here. So that's what the atypical is, forming this big blanket. The typical is doing something very different though. It's forming these weird kind of tendril-like attachments. And it seems to form this kind of desiccated structure as well. So our next question was, it's clearly making these things that allow it to persist for years just on these surfaces. And this probably is important as it relates to spread of the disease, right? For moving this around the apiary on our hive tools and on our bee suits. So our next question was, okay, it's persisting on wood, it's persisting on steel, right? How do we kill it? So we looked at a couple disinfectants. The first one was 70% ethanol. We wanted to choose disinfectants that were kind of widely available and generally considered effective on other bacteria. Um, so what did we find? We found on wood, it killed almost 100% of atypical and typical strains. There was still some bacteria that seemed to survive, but a lot more of the bacteria survived on steel for some reason, and we're not quite sure. But we do attribute these survivors probably to that biofilm and ethanol not being able to break it down. So we're trying to do some more research on what that biofilm is actually made of. But next we tried 10% bleach, and this was super effective. So it killed almost 100% on wood. It was like 99.999% or whatever. Basically, killed all the stuff on wood for atypical and typical. And then on steel, we didn't recover any bacteria after 10 minutes. So this was 10 minutes of treatment with ethanol is pretty effective. 10 minutes of treatment with 10% bleach is highly effective. Bleach got the gold star there. So this is kind of where we're at now. We're still doing some ongoing research on what this means clinically, what it means for, the hive. Can we decontaminate material and improve, decrease disease rates the following season? That's ultimately the question. And obviously, you can't pour bleach on your honeybee colonies, so we're looking at other stuff that would be food safe that might help us control this disease. So that's what we've been up to the last few years. That was like the basis of my thesis were all those kind of surveillance and antimicrobial resistance and then disinfection. And I think I'll end it there and I'll let Megan talk about how we manage this disease and kind of future directions. And then looking forward to questions from y'all.
All right, thanks, Peter. And hopefully I can still control everything. So I'm sure a lot of you are here because you've had EFB or thought you've had EFB or are afraid you're going to have EFB. So we're going to talk about um, some of the actual solutions. I will say that we have not solved it, and we don't have a silver bullet or quick scheme for this. So, but I'll give you some information that can help you make decisions. Hopefully. There we go. All right. Well, and also so that you don't have to panic and burn it all. So what we want to do is do diagnostics as quickly as we can, deal with it, and then move on with life. Honeybees are animals. They are going to get sick. It's also a really, really good opportunity to show your friends, show your neighbors, make sure that if you're working with a veterinarian that they can come in and see it. Like I said, we do have all these online things for showing you how to identify it, but it is very, very different when you see it in your with your own eyes. Peter and I are very lucky in that we get to visit thousands of sick hives a year. So we have a very clear sense of what it looks like, the range of what it looks like, all the different expressions of European Foulbrood. But it is really helpful to actually see it. And you can see, people wearing gloves and not then taking the disease frames and putting them in their own. All right, so the first thing that you want to do is confirm that you have European Foulbrood. I mentioned the diagnostic tools, which has the visual signs. There are lots of visual signs. One of the main characteristics of European Foulbrood is that it is pretty diverse. So American Foulbrood, you kind of see the larvae go through the same pattern as they die. Sac brood is kind of the same thing. Chalk brood, you always see the same thing. But with European Foulbrood, you'll see these tracheal signs, you'll see melted, you'll see the twisted larva. So lots of different types that you'll see. It's really spotty. You can see it affect the cappings, as you can see here. It can melt down and form a scale that looks very similar to American Foulbrood, but you'll be able to remove it. You also do, it kind of looks like we have some chalk brood in here. We definitely do see co-infections happen. We have seen European fall brood at the same time as chalk brood. We've seen it at the same time as sac brood. And we've also worked with a beekeeper where we had a lot of EFB and then it transitioned to parasitic mite syndrome later in the season. So you definitely can get co-infections. Oops. And then you get, again, as I mentioned before, you can go to the pollinators.msu.edu site. If you suspect you have it, send and you can upload some photos and you can ask a question and we can help with your diagnosis as well. Last week I was at the American Honey Producers Association and I was talking to the people that produce these lateral flow devices. There have been some issues with production for them. They should be back out this spring. So I was told March. They are a little difficult to purchase in that you can't buy them directly from the bee supply stores, but we are able to get them through grants at the university, and we're hoping to make them available to veterinarians through some projects that we have. There are ways that you can still access these, but there are these lateral flow devices that you can use for a positive diagnosis. One of the really, really key things that we focus on in our online diagnostic resources, and that is important for getting a positive response, is to make sure that you're testing larvae that are in early stages of diseases. So right here in the middle, you can see some larvae that have kind of assumed this corkscrew shape or have kind of become malpositioned but they still look alive. They're still kind of plump and juicy little larvae. They're white colored still. They haven't looked like they've started to decay. And that's what you want because all of the diagnostic tools are going to look for the presence of Melissococcus plutonius, and that will tend to get disappear as the other bacteria take over. So if you start to take a sample that is really heavily decayed, then you oftentimes will get a false negative. So this is true both for doing the lateral flow device and then also if you're going to send samples into Beltsville. Another important thing is to rule out Varroa. So I did mention we did have colonies we followed that had European fall brood and eventually turn into parasitic mite syndrome, which is when we start to see the damage from the viruses transmitted by Varroa. But if you can do an alcohol wash and, if you see something like this, it's probably not EFB. The other way to rule it out is going to be seasonality. We, as Peter mentioned, often see most EFB in June, and then we see most of the parasitic mite syndrome really in like September. However, we can see both earlier. There is a question that was asked pretty early on here, and it says, if you can clarify whether snotty brood or idiopathic brood disease is still considered a condition distinct from European fall brood, or is it simply an atypical form? So a snot brood or the crud or idiopathic brood disease was really talked about a lot, kind of around 10 years ago or maybe 8 years ago. And that was where people thought that they were having European fall brood or something, but it didn't look like classical AFB. It didn't look like classical EFB. And so, the term idiopathic brood disease literally means like brood disease, we don't know what's causing it. So it is kind of hard to tell what was happening there. I think there were probably 3 things at play. One is that was also the same time that we really started to see some of these larger effects from the Varroa transmitted viruses. And so a lot of it was parasitic mite syndrome, because you do often see really melty looking larvae and lots of brood disease signs in it. So one is I think people were thinking it was a bacterial disease and it was, mite derived in high mite loads. The second thing that could be happening is we are seeing different strains of European fall brood. So a lot of times people will talk about it looking different. And we talked a lot last year in our webinar about the different strains and the genetic diversity of the samples that we found. It is diverse and you can have other secondary bacteria be really diverse. So it can look different and it is variable. The third thing is that sometimes people were saying that they were seeing European Foulbrood, or they thought it was European Foulbrood, but then it would test negative. And I think what's happening in that context is that people were testing samples that were much too far diseased and were too heavily decayed, and then they were getting a false negative. So I think that the snot brood or the idiopathic brood disease, in my mind and in talking to other researchers and other extension people, it probably usually was either a false negative on European fall brood or it was the parasitic mite syndrome. All right. So I mentioned the strong seasonality that EFB, we would see it in the spring and summer. If it's in the late summer or fall, then it's going to be the parasitic mite syndrome. You can confirm your samples by sending them still at this moment to the Beltsville lab. So the USDA maintains a honeybee lab in Beltsville, Maryland, where you can send diagnostics. And on their website, every time I try to find the website, I just Google send bee samples to Beltsville lab. And they do have a lot of instructions that discuss that you can send a chunk of comb. But that only really works well if you are not using plastic foundation, because the reason that they request a chunk of comb is they also use visual identification. So you would send like a 2-inch, I think, 2 by 2 inch piece of comb or 3-inch piece of comb. And then Sam, who receives them, would actually look at the larva and that would confirm his diagnosis or be part of the diagnosis. But when you have the plastic, it's really impossible to cut through it in the field with when there's larvae in it. And so they do have instructions that you can take a swab as well. So you can take like a sterile swab and just take a larval sample. And then in that context, you would want to take a larva, again, that we discussed, that's kind of newly diseased. Or you could take a few larvae, but you want to make sure that it's the ones that are kind of still white and twisted. So as Peter mentioned, we have lots of inapparent infections. So we have lots of cases where you have the bacteria present, but you don't necessarily see the signs in the larva. And so we really do treat this like a herd disease. One of the things that you'll often hear people recommend is, quarantining individual hives. But when we see the sign of disease in one hive in the yard, it's often found in adult bees in the rest of the hives in the yard. So we call this very high levels of inapparent infection. There's lots of data. So I list a couple studies that show that in colonies that are located close to EFB, symptomatic hives, M. plutonius is highly prevalent in the adult bees and the larvae, even when there's not symptoms. And so you can see a whole list of studies that have found that. We've found that in our own research as well. And so one of the things that doesn't really make a lot of sense is to take that one colony that you see the signs of disease and then put it in another yard. And any beekeeper that's had EFB in their hives multiple years in a row will know that, if you have 5 hives, one year it shows up in hive 1, and then the next year that hive looks fantastic. And then in that year you see it in hive 3. So you can kind of think about it as the pathogen is kind of spread through that yard, or perhaps your whole operation by the time that you notice it. And then you want to think about, everything having the pathogen, even if you don't see the signs. All right, so options for treatment and control. We have done studies looking at just feeding sick hives pollen. We've also done preventative studies where we give the hives pollen and see who develops disease. And at least with the current formulations of pollen patties, the ones that we studied, so those studies were in 2018, 2019. We did not see any benefit of just feeding the bees pollen. And again, that's another recommendation you'll hear people say just make sure they have good health. And we can see colonies that have access to ample pollen or that, and like they are in excellent shape. They have no signs of disease. And then when the season hits, they will crash. So it's not just a lack of pollen, and giving them pollen supplementation does not work. We also have personally tested some different formulations of probiotics, and we don't have any evidence of ones that work. Notice though there is like an all caps yet on there. So in 2025, we don't have products that we can put in to either prevent or speed up the hive getting better. But this is something that we really hope comes, and we're going to continue to test them. Actually, even this coming year, we're putting in grants to continue to test products that are out there. But we don't have a recommendation of, oh, just put this in and it'll prevent the disease or it'll make them get better faster, except for the antibiotics. So as I mentioned, we don't have a silver bullet. It really is going to depend a lot on the economics. If you were entirely risk averse and you are in a remote location, you're not buying packages or nukes every year, then it might make sense absolutely to treat European fall brood like American fall brood. So there are some places in Europe where they actually do automatic burns for European fall brood just like they do for AFB. So I think that if I was a beekeeper that knew I was in a really remote location, I hadn't had it before, and I'm not buying bees, then it probably would make sense to destroy that hive. And I actually did have a beekeeper this year who I visited their operation, and they had caught a swarm, and that was they had never had a history of EFB, but they had caught a swarm and that had EFB. And we had talked about that would probably be a good place for destruction of just that single hive before it was something they had to deal with from year to year. I'm going to talk about the second one. So the antibiotic treatments, as Peter mentioned, do still work as a way to reduce the amount of time that the colony is sick. So it is something that does reduce the disease load. It is better for the health of the bees in terms of, healing faster. You can also do the shook swarm, which is a way to get rid of the amount of pathogen. You can also do watchful waiting if the disease is not incredibly severe. As we mentioned, that a lot of times or sometimes it does just get better, though that is something that we've noticed is not always the case. So if you have a colony and you just wait, it's not necessarily going to just get better. So here's the antibiotics applied as well. So the shook swarm method is a method that does have a lot of data for reducing the chance that the disease comes back that year, and then also the following year for that hive. It does not eliminate it. So I'll give it as an option, but it is a very expensive and labor intensive option. So the first thing that you do is you set up a hive with brand new clean equipment, and then you are going to shake the bees onto new equipment. You can see in the photo at the right that there is the sick hive is the taller one with the 3 deep boxes. And then we're going to shake those bees into the one with the blue box on the top. And you can see the blue box is totally empty, and that just acts like a funnel. So you can shake the bees in there, and then we would get rid of the box. And then you have to decide what to do with those frames. And I'll talk about your options for that. And then you can treat all the colonies in the yard with antibiotics. So this is the same method that we use with American fall brood as well. Actually, those photos were taken from colonies with American fall brood. The idea behind it is that you're getting rid of a lot of the pathogen. So all of those larvae, you're getting rid of everything that's on those frames. And then the bees that are put back in there, they have to, if they have the pathogen like in their mouth parts or in their crops, they can't just regurgitate it into drawn comb. They have to actually swallow whatever they have and either leave the hive to defecate or turn it into wax. And so it will be a while before you have larvae again. And then they also are going to have the antibiotics. So it is a way that is, it does really help a lot. I do want to reiterate, though, that in the scientific literature and in my own experience, it does not guarantee you'll eradicate it. With American Foulbrood, we've done this. I've done this multiple operations, and it's just gone. You don't have to deal with it once you do a shook swarm. It really does eradicate it or kind of solve the problem. With EFB, I've had it even come back the same year. And one of the main differences between the two is that the European fall brood is known to be in the adults. So we have some recent studies that came out from a really good lab in Saskatchewan that's been working on EFB for years, and they can show that the adults are affected as well. So with American fall brood, since adults aren't affected, you can take the bees, put them on new equipment, and as long as you have a gap between when there's larvae and they get rid of the pathogen, you kind of solve it. Whereas with EFB, you're going to put the bees on new equipment. You've definitely gotten rid of a lot of the pathogen, but those bees can have the bacteria in them. But if you are going the route where you want to try to aim towards eradication or really, really reduce the amount that's in your operation, you could set up, basically a quarantine yard after you've done the shook swarm. It is expensive because you often do get rid of frames. So I mentioned you can treat it like AFB and burn the frames, or if they're plastic, put them in a contractor bag and take them to an incinerator. This is what I do with colonies when I do shook swarms is I melt down the wax, but and sterilize the wood, but I do reuse the frames after they've been cooked for 5 hours at 500 degrees. But you do have to figure out what you're going to do with all of that equipment. You, we mentioned the bleach, and I do see some questions about all of these disinfections. I will say that in this moment, the Saskatchewan lab is working on disinfections. There's a group in Switzerland working on disinfections. And then our main projects this year is working on disinfections. So we don't have, so the work that Peter presented was on what we've done in the lab, where we put it on wood and then we cleared it off of wood. We haven't yet done the studies on what it works in the field. And part of the reason for that is because, basically we're mentioning how hard this bacteria is to work with. It's also really hard to induce infections in the field. So I can't just take a colony, spray some Melissococcus on there, and then automatically get disease, which is good. It means that we're not running around spraying everything with EFB. But it's a really hard thing for us to study then. It'd be nice if we could say when we did this, then we didn't get disease, even when we challenged it. So those are called challenge studies. So we are really heavily looking into disinfection and sterilization. The other thing that we've kind of been playing around with at MSU is working with an EFB yard. So one year we had another veterinary student, Alyssa, that looked into the effects of shook swarm on reducing disease. And, we have EFB, we've had it on MSU campus, and which I love because I can use it as a teaching tool. I think our farm manager probably loves it less than I do. And we are looking at ways that we can eradicate it. And so what we did is we did a shook swarm of an entire yard. Because as we mentioned, once you see it in one hive, it's in the rest of the hives of the yard. But we did that shook swarm in May, and a lot of the equipment looked perfectly lovely. And we had a lot of frames of brood that looked completely healthy. And I am not a monster. I cannot burn a perfectly healthy, good frame of brood. And I'm sure, if I can't recommend that to beekeepers, nobody's going to ruin perfectly good frames of brood. And so what we thought about is setting up a yard that was completely shook swarmed, got antibiotics. So that was kind of a clean yard. And then that equipment, if it didn't look bad, went to another yard that already had EFB. And so that was kind of a dirty yard. And so things could go into the clean yard, could only be brand new equipment. And then the next year, you could take another yard and do the same thing. So the idea is you're systematically reducing the parts of your operation that would still be at risk for having EFB. So you can also just kill the colonies. These are the guidelines that are going to be coming out, I believe in January from the American Veterinary Medical Association for their depopulation guidelines. And they'll have more details for that, but you can just use an insecticide. There are some insecticides that you can use and reuse the equipment, which doesn't necessarily make sense for EFB all the time, but and then if you're not planning on using the equipment, there's lots of recommendations on how to use diesel fuel to kill the colony. So I mentioned the shook swarm isn't going to be perfect. Even if you euthanize one hive that has signs of disease, that's not necessarily going to be perfect because the pathogen is going to be in the other hives in the yard as well. So the shook swarm can be used if it's early enough in the season so your colony has time to rebuild. If you want to sell nukes from that yard in the future, it is something that I think you do really have a very strong obligation to try to have the newest equipment in there as possible and to minimize the amount of pathogen you have in your operation. And even better if you can kind of shook swarm a yard at a time. If you're going to do it at the larger scale, I kind of mentioned this a little bit, but you can have these kind of clean yards or dirty yards and making sure that you have equipment flow in one direction. And to think about how you do comb replacement. So this is another project that Peter and I will be working on this spring. We already have funding for is working with beekeepers who, instead of just taking a hive and splitting it into two, and occasionally just replacing the occasional frame, is when you take a hive, they're actually shaking bees onto new equipment. And so you kind of have an old hive or a parent hive and a daughter hive that has new equipment. And so you're always thinking about how old your equipment is. And you would call from that old hive and make sure that the new ones have new equipment. And I'm going through these fairly quickly, but I'm happy to do follow-up questions, or if people want to reach out, if they're actually interested in doing these, or if you have it in your operation and you want to do this in the spring, I'm happy to work with people one-on-one. So the antibiotics are the other option. Where those really fit in is when we're trying to control disease. So if you are in a place where you really just can't switch out all the equipment, which is especially true for people who are not managing mites sufficiently, so they're still buying bees. So if you're going to import a nuke, if you're going to buy bees the next year, it does not make sense to be drastic about EFB because it's so widespread, you're likely going to purchase EFB. And that doesn't mean that the bee suppliers are just selling hives knowingly with EFB. As we mentioned many times, you can have a colony that looks perfectly healthy, but the pathogen is present. So, in the other way that it wouldn't be useful to do these drastic measures is if you're in an area with lots of beekeepers, which in Michigan, most of us are. If you've got a colony that requeens itself just fine, that means there's other bees in the area. And so through robbing and through things like that, you can actually pick it up as well. And so a lot of times we're not trying to eradicate it from our operation, we're trying to control. So we're trying to minimize the damage that it does within the hive. We're also trying to minimize the spread within the hive, minimize the spread among the hives in the yard, and then especially between other yards and other operations. And so that's where the antibiotics do become useful. Oops. You will have to work with a veterinarian in order to access antibiotics. They will have to write you an order before you can purchase it. We have a lot of training programs for vets. So if you are a veterinarian, you can access these here. If you're working with a veterinarian and they say, oh, I'd love to help you, but I don't know anything about bees, I can work with them directly. We can make sure that they get to the point where they can feel comfortable writing you an order without worrying about losing their license. And then there's lots of resources there for them. We also maintain a list of veterinarians who are working with beekeepers in Michigan. And so it shouldn't, and if you can't find one, I can help you in Michigan come into contact with a vet. So, as Peter mentioned, we only have one antibiotic labeled for use in EFB, which is oxytetracycline. There are other antibiotics that have been shown to work in the lab, but at the moment, only one is labeled. So, terramycin is the one that we use, whereas Tylenol and lincomycin are effective in the lab against Melossococcus. You have to get an order from the veterinarian to access this. They can write you a VFD, and if they do that, they'll send it to one of the bee supply stores like Man Lake or Dadon's, and then you can just order it directly from the store. They may also be able to write you a prescription, in which case you can get it from a pharmacy, or ideally that veterinarian might be able to provide you access. And then you do have to follow the instructions for the application. And especially paying attention to the withdrawal period. So here's the tough thing about it is that the antibiotics can only be used when the hives are not collecting honey for human consumption, and in fact, 6 weeks before that. So you can see that you would, not be using it on a colony that is actually actively collecting honey. We have used it like MSU on small colonies that are not collecting honey early in the season. So you have to think about when you're going to put your honey supers on, and you can do 6 weeks ahead of that, or when you've got equipment that's not used. So if you've got a colony that is growing and you think it's going to get too big that it needs more space, you can add a third deep and then not use honey supers on there, and then just know that you're not going to extract those deeps for human consumption. But it is really tough because when we start to see most of the signs of disease, which is June in Michigan, that's our big honey collection period as well. All right. And I do like to point out the term metaphylactic use. So a lot of people talk about antibiotics and they don't want to do prophylactic use, which is really important because we do want to have judicious use. But we also want to make sure that we are not having animal welfare issues and not managing disease in our hives. In the context of honeybees, once you have seen the disease in your operation, it's going to be in your operation for many years because it's on the equipment. And we talked about like all of the inapparent infection. And so in that context, it's not prophylactic use where you're preventing the risk of something happening. It's under the term of metaphylactic use where we're controlling the spread of a pathogen that we already know is in our herd. And I like this photo in particular. It's a little dark, but you can probably see this is from almond pollination. And you can see that there's multiple different operations here. And again, with European Foulbrood, our focus is really on control. So, when we've got big operations, even small operations, we may not be able to eradicate it. We just really want to minimize its spread, and we want to minimize how bad it is. So if you know you have it within an operation, it is appropriate to use the antibiotics even before you see signs of disease from that year. And I do have language from the FDA to that effect. So you don't, if you have had European Foulbrood every single year for the last 5 years, you know it's going to pop up. You don't have to wait until the disease is bad before you start looking at antibiotics. And the reason is because we know that pathogen is already circulating in your operation. All right, so we don't have good answers for prevention. The best thing that you can do is prevent it from getting into your operation at all. So I mentioned that we've tried doing nutritional support. We've tried looking at different strains of hygienic bees. We are continuing. We've looked at some other ones too. That's something that would be a lovely long-term answer. We have played around a little bit with the vaccines. They aren't enough to prevent it completely, but we've not done a formal study. And really, it is because it's so widespread. So if you're going to be bringing in bees, catching swarms, buying nukes, buying packages, it is in a third, 1 out of 5 colonies. And so eventually you will get it. And even if you're buying a package and not buying the frames, and they look perfectly healthy, it could still be in the adult bees. So the sooner you can close down your operation and not buy bees, the better. You could also think about biosecurity and the things you're bringing into the yard. So we use nitrile gloves when we go to other yards and even in our own yards. If you do have to use leather gloves, think about 1 per yard. Same thing with smokers and hive tools. The smokers are, or hive tools are pretty easy to clean. Clean everything often with bleach and really think about where you're moving your equipment. It is pretty natural to mix everything together, but the more kind of boundary systems you can set up, the better for prevention. All right, so main points is we don't yet have a great system to completely eradicate it from your operation once you have it. Shook swarms absolutely do reduce the pathogen load, and if antibiotics reduce the time to recovery, so we can use those to control spread. And there are people that have had pretty good success or multiple years without having disease, especially when they combine the two of those. Just keep in mind that this is a yard level disease. So if you see the signs in one hive, you can likely find the pathogen in many hives in the yard. And we're doing a lot more work right now on kind of strategic hive equipment and movements of your equipment. All right, so now we will work through some of the questions. I haven't pre-screened them, so we'll just kind of go through and we'll stop when we either don't know anything anymore or everybody gets so tired. So, um, Peter, there is a question about survivability or what we know about the survivability of Melissococcus on wax and in pollen and honey. Do you want to address that? And you're muted right now.
Oh, sorry. Can you repeat that? I was unsharing the cursor so I could actually unmute myself and stuff. It was kind of
Yes, exactly. So the
complicated.
question is, what do we know about the survivability of Melissococcus plutoneus on wax and in pollen and in honey?
Yeah, not a ton. People regularly culture it in studies out of honey. We've tried to do that in our lab and haven't been very successful. Um, as far as it being in wax and what was the other one, pollen, I
Pollen,
know pe
yeah.
people have cultured it out of those materials, so we know it's alive on those materials, but we don't know the concentration. So we don't know if it's at the level that that's what's causing spread. And I think that's the next kind of big question is it's probably alive on there, but is it important, you know?
Yes. And a similar question, I'm going to try to look at some persistent ones. Can the bacteria survive several weeks of freezing temperatures?
Yes, it definitely has no problem in cold temperatures.
Yeah, we can.
That was one of the things we tested also with Robin's study was she froze some stuff and we grew it pretty readily after freezing for quite a while. So
Yes. And I mean, yeah, I've grown it out of samples that have sat in the freezers for years. And that's where we store it is in the freezer. So, unfortunately, winter, it would be nice if winter helped us with that.
yeah.
Um, all right. So so there are some questions that are like I mentioned, we're starting down the path for disinfectants, and this is the danger of presenting research, while we're still working on it. We do not have so there's a question for 10% bleach for how long do we spray it? Do we soak it? We have, we did 10% bleach for 10 minutes, but we were doing little dowels in the lab. So
Yeah.
we don't have, and what we did was show that we can get a reduction at that, but we have not done the studies that translate that of what's in the field. And, we really want to give good recommendations on how to prevent disease, how to treat your equipment, but we also, for us, even getting negative results are useful. We don't want people spending a lot of time and money doing things that don't work. Because a lot of times, especially in extension or even as a veterinarian, you would want to, you want to help people. So you want to come up with answers. And one of my biggest frustrations is there's all of these things on the internet that say, oh, just feed them and they'll come out of it. Once the honey flow turns on, they'll come out of it. And that's just profoundly not true. And so we want to make sure that when we're giving you recommendations, they actually have data behind them. So where our lab and there are some other labs that are working on some of these, like we're aiming to be able to give you a guide that says, this is how you would do it in a way that's useful for your equipment, but we are not there yet. And there is another question about fumigation with acetic acid. That is on our list of one of the things that we are planning to try this upcoming year. So we do have a list of things that are available to beekeepers, safe for beekeepers to use, and that would potentially work that we're planning on testing.
I will say we did some preliminary stuff when we were doing our disinfection assay. Uh, we did a couple rounds with vinegar, a couple rounds with UV light. We didn't mess with the dosages or anything. We just did what was readily available, vinegar you buy at the store. And we didn't find any reduction in bacterial loads for the vinegar, and we didn't find any reduction from the UV light, at least at the doses that we did. So, just wanted to throw that out there also for people
And
who,
hydrogen peroxide.
yeah, we did try hydrogen peroxide, and that didn't seem to work either.
But
So,
Peter brings up a good point too. There's almost unlimited variables of time and temperature and conditions. And so, um, another point that he did not stress enough of how hard it is to actually quantify this bacteria and quantify the amount that's alive. So these disinfection studies are actually a lot harder to do. Um, than you think. So there is a question that says, have we tested any wax comb after the bees have cleaned out the disease cells to see if EFB is still lingering?
that's an awesome question. I totally want to do that. Like that is like one of the top questions that I have is how well they're cleaning this out. Yes.
And we did some like very early stuff just poking around on dead out colonies that we knew had EFB and old equipment. And I was having this conversation with someone today too. It's hard because there's so many different places you can test. Like if you test bee bread, but you test, one cell, they have 300 cells of bee bread, one cell could have it and another one doesn't. Or the
Yeah.
same thing with the cleaned out cells. We don't have a sense of how variable or how spread it is throughout the hive. But yeah, that is a good question. Um, here's kind of a disease one. So it says, um, and I'll let you start with this, Peter. With the prevalence of EFB in the hive at times of the year, does it have any impact and synergistic effect with other pathogens or viruses that could result in the hive's demise?
Yeah, that's a good question too. We have noticed there seems to be some association with sac brood, and we did put out a paper on that. And we also looked at deformed wing virus, and we didn't notice any correlation with deformed wing virus. But sac brood also tends to have a similar seasonality, as does chalk brood. And we do see both those diseases showing up in EFB affected colonies pretty commonly. And maybe that's just because they have the same seasonality, but not a lot of research has been done on how these might be working together. So I think that is a really good question, and I've wondered about that often myself.
Yeah, and to build on that too, with the we really, really focus on the role in the larva and how much disease we see in the larva. But the group in Saskatchewan this year and just put out that there's a survival effect in the adults as well. So you do see reduced survival in the adults. So it's probably worse or doing worse things than we anticipate when you're thinking about at the hive level or when you have EFB in the background and then you get, a nutritional stress or a pesticide stress. It does make sense that you could see some synergistic things, but there definitely isn't a lot of data. Um, all right, so there are two questions about passing the hive tools through a flame, if that's effective in the field, and then also passing it through a flame with ethanol. Do you want to comment on that, Peter?
Yeah, I mean, it's my opinion that getting things off of the tool is probably the most important because I think even with American Foulbrood, they showed that when people flame boxes, if you go a few layers deep into the box, you can find viable spores. And so it's tough to say if you're killing everything, if there's a bunch of gunk on it, if you're just flaming it. But we do know off of some 100-year-old research that G.F. White did. So this is like in 1912 or 1920, I think. Um, he did take diseased larvae and then put them through different environments, including heat, and he established sort of a heat death, at least for the pathogens he was working with. We don't know what strains they were or anything. This was a long time ago. And it was a relatively low temperature too. So I think we haven't done the studies recently, but I would suspect that heat is going to be a pretty useful thing. But that's again based on 100-year-old data. So another question
And
I have.
yeah, and the thing with the flaming is it's so usually people are so quick with it. And
Mhm.
usually when you're using heat to sterilize, it's temperature times time.
Yeah.
So you can do a really high temperature for a short amount of time or kind of a lower temperature for a longer time, but there is kind of both of those are important. And a lot of times people are so quick. But as Peter mentioned, getting the stuff off the tool is probably the most important thing. Our protocol that we use for our hive tools is that so and I do this now in my own operation after realizing how gross I probably was with my own hive tools, is we do have many of them and we just have a tub of clean hive tools and then we have a tub of dirty hive tools. So if you're in a hive that looks suspect, you can just put it in the dirty one, or what we do is try to do every yard gets a new tool. And then with the dirty ones, those are in a bin and we put them in bleach water. And we're in the field, so we're not measuring, but it's probably around 10% bleach. And we just let them soak in the bleach water. So you get that time element. And then we'll use a scrubby and like Comet or Barkeeper's Friend or a scrubbing powder. And we'll just actually make sure that everything is off the tool or use two hive tools against each other. And you make sure the tool is totally clean. And then we rinse it and then it goes back in the clean pile. So we do put quite a bit of effort into the hive tools. There is a question that says, "Are you destroying the frames?" And I think that was probably when I was talking about the shook swarm method, and I believe this came in probably before I kind of went through the options, but you can destroy the frames. That's just really expensive if you don't think you have a good chance of eradication. So you can destroy the frames if you think you can eradicate it and or just if they look bad or if they have a lot of infectious material. So when I do the shook swarm or partial shook swarm where I take out the disease ones, I do tend to throw out the ones that, or in my case, melt them down, the ones that do have lots of disease um, larvae. So you know that there's a lot of pathogen in those disease larvae. And if you don't take them out, the bees are going to use their little mouths to take them out. All right. So um, there is a question about how detrimental the colony is having. So, do you think ongoing EFB in the hive going into winter would have a detrimental impact for colony survival? If so, what would be the best way to prepare a hive for overwintering? We have seen colonies with signs of disease at least through September. We don't tend to monitor past that. Um, there isn't, I mean, if they still have brood disease, it's really hard for them to have enough bees. Um, when I had a yard that um, had quite a bit of EFB kind of late in the season, usually what you end up with is just lots of small colonies. And so what's often recommended in that case is just to combine them. And so that everybody, instead of lots of small, because the issue is that you just don't have enough winter bees getting raised. So the best thing in that case would be to combine them, or if they're really small, then you would at the end of the season think about euthanizing those colonies. There is a question, is it possible to get the test kits anymore? I did touch on this. They I did talk to the company literally like 3 days ago, and they will be available in March, but you have to buy them directly from the company by the case. So you can't just buy them from a bee supply store. But if there's veterinarians, especially in Michigan, or people interested in Michigan, we're hoping to work on some projects to bring them into the state. Um, all right, so there is a question, how do we prepare a sample for diagnosis and send it in an acceptable way to the carrier? So I will tell you what I'm going to do right now, real time, is Google send bee samples to Beltsville, and the bee research lab comes up, and I'm going to put this link in the chat, and then I'll also put it to answer this question. Um, And the big thing is that they don't want it to be like really wrapped tightly. They want to make sure that it is not going to just rot in there. So, if you're sending brood samples, you don't want to have honey in the sample. You can send it in a paper bag or loosely wrapped in a paper towel, newspaper, etc., and send in a cardboard box. And they say avoid in big letters wrapping such as plastic, aluminum foil, waxed paper, tin, glass, etc. Also, these instructions, I found a article from the 1930s that had this like word for word the same, which is why they talk about like don't send it in glass. Like, and my joke is that the USDA hasn't updated their website since like 1934. But and then it says if comb cannot be sent, the probe used to examine disease larvae in the cell may contain enough material for tests. The probe can be wrapped in paper and sent to a laboratory in an envelope. So we buy sterile probes that are basically Q-tips. And so you could use a Q-tip, dig it in and get a couple of those larvae, and then just wrap it in a paper towel and put it in a envelope and send it to them.
I would say to emphasize to avoid the urge to put this in a plastic bag. Because like you want to put it in a plastic bag. It seems like the right thing to do when you're sending it in the mail, but you don't want it in a plastic bag because like it'll mold and like it's impossible to read the sample once there's mold on it. So that's the whole point of like the paper and the plastic and the cardboard and all that stuff to let it breathe a little bit.
Yes. Um, okay, so there's some more questions on the disinfections. So Peter, can you please clarify how you used the 70% ethanol and 10% bleach? Did you soak? Did you spray it? Did you
Sure.
think that spraying would work instead of soaking the hive boxes?
Yeah, so what we did was we basically inoculated a dowel, a wood dowel with this bacteria, and then we would let the bacteria dry down onto that. So the bacteria was on every surface of this. And then we would dip that in ethanol briefly, and then we would let it drip dry for 10 minutes. So it was kind of coated in ethanol. And then after 10 minutes, we would rinse it, and then we would pull as much bacteria out of that rinse as we could, and see what the quantity was compared to the original. So that was kind of the process, if that helps. But we were hoping to mimic what it would be like to spray, but spray is like tough to quantify in a normal way, and so this was kind of our solution.
Hmm.
But I think like moving it out of the field and actually practically doing this is kind of our next step. So hopefully we'll have better answers in the future.
And we're also looking into some techniques that are really like fumigation based.
Yeah.
So the idea is you could put all your equipment in a trailer, do some things, and then it would be safe afterwards. So there's a question that says, how long does it take for the samples to come back? I'm guessing that's from the USDA lab. It really depends on how many samples they're processing and whether or not the government is shut down. Um, I do want to say, so we are getting set up to take samples at MSU. We're not ready yet, which is why I'm not going to talk more on that till there will be huge announcements when we are. But the Apiary Inspectors of America also has a list of labs. So I'm going to Google that really quickly for you. And they have on their website a downloadable PDF of where you can send samples. So the nice thing about the Beltsville lab is it's free. The downside is when we do have government shutdowns, it's not available. So I'm going to put this in the chat right now. Um, oops, I didn't hit the rest one. So there's how to send samples. And then the AIA has Find a Labs, and they have a listing of all the diagnostic labs that also does list labs that will take pesticide samples as well. Um, all right. So I'm just making sure. With both Foulbrood diseases, have you ever noticed a definite smell or stench? Peter, do you want to talk about your hives and their names, perhaps?
Oh, Smelly Melly and Clive the Hive. Yeah.
Mhm.
Yeah, the European foul brood definitely has a smell, but they've associated that smell with one of those secondary invading bacteria that's not always present in the hive. So you can definitely come across it without the smell being present. But when the smell is present, it is like very present. It reminds me of like sort of, it smells sharp, I would say, vinegary, like tart-ish, pickled sort of material.
Mhm.
But I mean, Megan, you've mentioned being able to smell it just in the driveway sometimes, even
Yeah,
outside the hive. So it can be pretty strong when it's present. I mean, but you're smelling just a bunch of rotting larvae, right? It's
yes.
very different from American foul brood, tends to have a more subtle smell, in my opinion. But I haven't come across as much European American as European. But the smells
And
are very different for sure.
the smells are very different, but in both cases, not every hive will have a smell, and also not every person can smell it,
Yeah.
and other things smell. So it's kind of one of those things that it's there, but it's not constant. There is someone that said that they've been a beekeeper for 6 years and have not observed recognizable EFB. Is this possible? Yes, absolutely. So it is. I mean, we're talking about how bad it is, and in terms of diseases, it's horrible. But probability wise, you may not have it in your operation, especially if you don't have very many colonies and you're not constantly buying them.
Mhm.
And I know a lot of beekeepers and it is also highly regional as well. So, we have a very Michigan focus. It's really bad in Michigan. There are other parts in the country where it's hard. We send them samples because it's hard for them to find it for their research. Um, but yes, it's absolutely possible not to see it, but it is one of those things that we like to make sure people know about because it's so widespread. That it's likely if you have enough hives for a long enough time and you're not and you're interactive in buying equipment and buying hives. But if you have a pretty closed operation, then it isn't necessarily that it will show up. There is a question about blueberry pollination contributing to EFB. I'll talk about it briefly. I've been to 3 meetings recently where this comes up a lot. Um, but there is association where people will say that honeybee colonies in blueberries get European Foulbrood. And this goes way back. So there was someone at MSU who did their 1983 thesis on high incidence of EFB after blueberry pollination. This is something we hear about as an issue in Canada, in the US, in the Pacific Northwest, in the East Coast. So it's not something new. It is not really obvious what or if something is happening. So when you talk to beekeepers, they definitely say blueberries are the worst. But one of the things that's loaded in there is that blueberries bloom at the same time that EFB peaks, or like right before. So they look really bad when they come out of blueberries. We have absolutely seen colonies that have nowhere been near blueberries, like our ones on campus, or backyard beekeepers or commercial beekeepers who don't do blueberries, that still get sick. And our own data show that if you take take a colony, split it, one goes to blueberries, one doesn't, we can still have high rates of disease. So there's a lot of data and a lot of stories that, bees do get lots of EFB around blueberries, but the understanding of why is not clear. And Peter, if you want to talk more about it, it says someone asked, oh yeah, like if a large percent of hives have EFB, what stressors cause it to basically flare up?
Yeah, it's a good question. Yeah, this is stuff that keeps me up at night. Like, what is it that, like, there's so many highs, and what causes it to go away, right? Late in the season. Why does it come on in early spring and then peak in June, and then the signs tend to go away? So, like, there's something there that I think could help us really solve this, but I just don't, the research just hasn't been done. Like, no one's formally looked at this.
Yeah. And it is, I mean, this is, I kind of jokingly made you answer that because I knew what pain it would bring you.
Yeah.
This is literally the things that we talk about till we'll get to the point of, is EFB even real? Like, why are we
Just
here?
in my head.
Um, it is really frustrating. The types of studies that we do, like we've done these studies called prospective cohort trials, where you take a set of colonies and then watch what they do over time. So we have a lot of longitudinal data of just following colonies over time. And there was one year that in order to be enrolled, they had to be perfect. And on average, they had 14 frames of bees. They had to have no sign of disease. They were queen right. And then like over 50% of them got sick. And this was while they were on a place where they were making honey. There was lots of pollen coming in. It was just, it's really frustrating because, I have hives that have bacterial disease, and they're on a 1,500 acre um, conservation area with lots of food and no pesticide. It's just it's really hard to figure out what kind of triggers it, or some researcher we were working with called it like tickled it to life. But we really don't know what makes it turn on. But that is a really good scientific question, and that's really what we're um working for. Um, there's a question about, did you use undiluted bleach for your 10% bleach NaCl?
Yeah, I think it equated to like 0.5 percent sodium hypochlorite, but the it was the standard what you can buy in the store diluted 1 to 10 or 1 to 9 to make a 10% bleach solution. We wanted to mimic like what people would do, like, with what they have at the grocery store. So like make this as widely accessible as possible is kind of the thought behind that. So
And then there is a question if that was the lowest percentage that was effective.
we only tried one concentration. We just went with the concentrations that have been recommended for other disinfectants, for other bacteria. Um, each one of these trials is extremely tedious because, like Megan said, the recovering the bacteria from these materials is very difficult because the bacteria is so difficult to grow, even though it seems to also maybe be difficult to kill. So it's an odd one.
Yeah, so I mean, it persists all over, but the thing that makes it actually start to replicate seems to really only happen in the larva. And even when we do our best conditions in the lab, it grows begrudgingly at best. It's still not growing that well, even when we're using the best methods that we know. But we do have a student that is focusing on that specifically for this upcoming year. Um, so there's a question that says, it sounds as if antibiotic treatment just suppresses and does not kill the bacteria. Thus, you have to treat every year and all hives in the yard. Um, and so, no, not necessarily. And we did a survey, or I did a survey a few years ago talking to beekeepers that have had EFB and what they use for antibiotics and what their history of use has been. And I didn't find a single beekeeper that just treated every year everything. And part of it is because it's expensive and labor intensive. And it does seem to vary by years. So some years you have good years, and a lot of times too, it seems to be that like if you have a really bad year with it, the next year's not quite as bad. And maybe it's how you make the splits. So I don't know people who use antibiotics on every hive every single year. Not saying that maybe somebody doesn't, but usually people will tend to use them when it doesn't seem like it's working or it seems like it's been getting worse. Um, I have used antibiotics. I definitely have not used them every year. We have used them on campus. We definitely don't use them every year. It really does depend on, how bad it seems to be. There's a question when you're using the shook swarm, do you treat immediately with antibiotics? Especially in the case of American Foulbrood, yes, but when we've done it for studies, we do treat with the antibiotics basically right away. And then it is a 15-day treatment. Um, if you discover EFB late in the season after the bees are not building wax anymore, how would you manage that hive apiary, assuming that a full shook swarm isn't an option at that point? Do you treat with antibiotics, remove the worst brood frames, and plan to do a full shook swarm the following spring? So that is a good point. If it's late in the season, the shook swarm's kind of off the table. You can use antibiotics at that point. You can combine it with another hive with from the same yard if you want. I wouldn't do the partial shook swarm is, where you just remove the brood nest. I've had it come back after that the same year. I would probably wait until spring. And then you can do a shook swarm in the spring. However, as I mentioned, it's amazing that sometimes that same hive will look fantastic in the spring. And then you have to decide if it's, worth it to do that effort on a colony that is, looks perfectly healthy. Um, so someone did ask about like prophylactic or automatically treating nukes or packages. I probably wouldn't automatically treat nukes or packages because again, you're only dealing with the antibiotics with what is happening at that moment, but then the antibiotics wear off and the bacteria is still there. So it is really something that we're using when we know that it's in the operation. There are two questions about whether or not you can get EFB from buying queens with attendance. We just submitted a grant on looking at the role of queens. I mentioned that the role of adults is just barely studied. The role of queens and drones is studied even less. So I don't know of any data that say that you can. I mean, theoretically, the attendance could have it, but I just don't know. I don't know if Peter, if you've come across anything.
Yeah, I know, like just mentions in those old USDA papers of like the queen is susceptible, but like it's a vague statement in these old papers and no formal studies have been done, which is weird.
Yeah. Yeah, the EFB research had its heyday about 100 years ago, and then a big gap in between. So there's questions about if you can put them in with sterilization. I mean, we really want to understand how to sterilize frames and how to do these, but we just haven't done them yet. All right, Peter, and I'm just going to answer questions. We're getting towards 9 o'clock. There's about 7 left. I think we'll just work through them and, a lot of people have left off, but people can see this on YouTube later too. So is it known how the disease is transmitted? It seems odd that it is difficult to infect a hive, yet 1 in 3 has it. Peter, do you want to start with that one?
That is a tricky one. I mean, when we look at the bacteria on these surfaces, we can see it's doing all this weird stuff, right? And if you listen to our previous podcast, we also know that there's all these different strains, even though there's two kind of like typical and atypicals, these two genetic groupings, there's all these subtypes within those. And we haven't really studied what these things are doing in the environment. So it's kind of unclear how they're persisting on equipment. And again, some of these strains, like I mentioned in that in vitro larval assay, some of them don't seem to cause disease in the laboratory.
Mhm.
So how they're working together in these co-infected hives, whether one strain's protective or whether they're competitive or whether they're working together to trigger virulence, um, that's something that hasn't been answered. I think that also has been complicating people's attempts to artificially make hives sick, for research also.
And another thing just goes back to the fact that like we can grow it, but under duress. Like it's not happy when we're
Yeah.
growing it. And so, it either loses virulence or it's not. We just can't replicate what it's doing when it's growing happily on its own. And that is probably like a big part of why nobody has studied it. And then also, why it's, because nobody studied it, we don't know some of these questions that are really well answered for other bacteria.
Yeah, when we
Um,
grow it in the lab, it looks very different from what it looks like when it's infecting a larva. So it's behaving in a very different way when it's in the hive than when it's in the lab. And whether we're selecting for certain strains, we're also not sure.
mhm. So Peter, someone has told me I might be able to look for the bacteria myself under a microscope. Did your prior webinars discuss how we might use a microscope, or do you think that it's impractical for a small scale beekeeper? I have a microscope.
Oh, yeah. If you have an oil immersion lens, if you have a 100x or a 1000x, it would equate to, you can totally do this at home. It's a very simple method. It's called the modified hanging drop. And if you go to that diagnostics page that Megan linked earlier, there's actually a whole walkthrough on how to prepare a modified hanging drop and how to identify Melissococcus plutonius under the microscope. And like you saw in those cross sections, when these larvae are infected, they're packed with this bacteria. So if you can get good at preparing these slides, it's actually pretty easy to identify this. This is how they identify it at USDA lab when you send in samples too.
Hmm. And we've done clinics for veterinarians a couple times a year and for vet students, but it is something you absolutely could do. Um, I love this question. Has anyone attempted trading a dog to smell Foulbrood? We actually have. So Peter and I are on a grant that was funded through the Michigan Alliance for Animal Agriculture, working with a dog handler who is working with American Foulbrood right now, which we do think is so that has been done in Maryland. They had a dog that was trained for that, working with their Department of Ag. We're working on writing a book so people can actually train dogs to do it more widespread. So we are working on it. There's a question if we're seeing yearly variations or if it's just constant regardless of the weather during the early season. So when we're working, so we've worked with some of the same operations multiple years in a row, and it is different every year. I would say, even just, with the same operation that kind of does the same route. And I don't know if it is because, so there could be a genetic component. So as the bees change, there could be something. There could be a weather component with so Leslie Bailey, who did a lot of the early research, talks about hive demographics being a trigger. So the ratio of open brood to nurse bees. And so that is something that is really closely linked to the weather. We don't know if, I mean, with the blueberries too, the pesticides can be involved. But the what makes it bad one year compared to the previous year or the next year is something that we just do not know at all. Um, Another thing that could be happening, which is linked to there's a question about the why we don't have a vaccine for EFB. So there is a vaccine for AFB, and the way that that one works, it is just a fixed kill bacteria, and bees don't make antibodies, but they do have a thing called immune priming. So when they get in contact with it, the idea is it's kind of like an elevated immune response, and it is a kind of general response. And the idea is you feed it to the queen, and then she passes that protection down through the eggs to those workers. It does seem to work, or there's some nice data from the people who developed it for AFB, and it doesn't seem to work for EFB. But there could be some sort of dynamic like that within the hive where the colony is exposed and has some sort of immunity so that the next year it doesn't have such a big response. But we really don't understand it. All right, and so there is something about whether or not humidity can be a factor for the trigger. Um, we don't know that it is. We're also looking at like CO2 levels in the hive might be something. So um, there are a lot of things to look at. And of course, they're all happening at the same time. Um, all right. So and then there's the last question says, I thought that atypical EFB what was seen later in the season. And so there is atypical Melissococcus in terms of like what we use in science, and that's based on, or what we use in research, and that is based on how it grows, how it's virulent, and like the genetics of it. So it is a different pathogen. The beekeepers also use the term atypical when they saw it acting differently. And that was kind of around the time with the idiopathic brood disease and the snot brood and things like that. And they noticed that it seemed to be hanging on longer. So there is like it's confusing because the word atypical was used in two different contexts. All right, probably should have answered that one sooner in the list, but I was going down as we saw them. All right. So I just want to thank all of you that quite a few people stayed on for a 2-hour presentation, or they fell asleep and forgot to log out. But I want to thank everybody for all your attention, and really, really good questions. And like I said, please don't hesitate to follow up through the website or ask us questions. If you want help with the diagnostics or treatment options or things like that. And hopefully next year we'll have lots of answers on everything, and we'll just have it all solved, and we'll have a really fun webinar last year. All right. Good night, everyone.
Bye.